as I was saying, the large intestine has these characteristic pouch-like structures, and that's due in part because the outermost longitudinal smooth muscle layer of the wall of the large intestine is reduced to just these narrow bands. And there's 3 of them. You can really only see 2 in total here. But these strap-like structures are called the tinea coli. And they're the reduced longitudinal smooth muscle layer of the large intestine. And because of the contraction of that muscle, it helps cause these pouch-like structures that are called the haustra. So the haustra are characteristic of the large intestine. Okay, so let's move on to the histology. No villi. That would be in the the villi are in the small intestine only. So in the large intestine, there are no villi. There are intestinal glands that you can see, similar to the gastric pits in structure that we saw in the stomach. There's abundant goblet cells that produce mucus to help the fecal material as it's produced to glide along. And notice all these little white areas in the epithelial lining are the abundant goblet cells. So notice how everything's really packed in together. With villi in the small intestine, there's more space between the villi, the structures that you'll see on the slide histologically. With the large intestine, you don't see those kind of gaps between these structures because these are not villi. These are just, invaginations into the epithelial lining, these intestinal glands, and characteristically there should be a lot of goblet cells. All right? Okay, let's get into the accessory gland organs. They facilitate digestion by adding secretions like saliva, salivary glands. Then we have the liver, gallbladder, and the pancreas. And we've talked about these a little bit already. So the liver, huge organ, relatively huge, right? About 3 pounds. It's our largest visceral organ. It has a lot of functions and only a few that we'll consider. Right upper quadrant of your abdominal cavity or your pelvic abdominal cavity. It does have over 200 functions. Importantly, metabolic regulation. It helps regulate circulating levels of carbohydrates, lipids, and amino acids. Stores excess glucose as glycogen. Remember glycogen storage in our liver, also in our skeletal muscles. Stores fat-soluble vitamins A, D, K, and vitamin E. Detoxifies drugs and toxins that we may have ingested. When you do have a meal, right, and the nutrients get absorbed into the bloodstream, the venous blood supply draining the small intestines and large intestine head towards the liver so that the liver can detoxify any potentially harmful items in the food that we eat. And then, of course, bile production that aids in the digestion of lipids through emulsification of the fats or the lipids. I mentioned that already. So the blood supply to the liver. Here's the liver, and its main blood supply is via the hepatic artery. Here it's called the common hepatic, right, off of the abdominal aorta. There you go. So it's supplying nutrients and oxygen to the cells of the liver, the hepatocytes. Again, hepato for liver. And then, so that's the arterial blood supply. Now, the liver, in terms of dealing with the nutrients absorbed from the digestive tract, has what's called the hepatic portal system. So this is blood that goes to the liver from the stomach, spleen, pancreas, and the intestines, small and large. Here, notice that the small intestine has been greatly reduced and some of the large intestine has been removed just to highlight this venous blood flow to the liver, the hepatic portal system. So the only ones you need to know here are going to be the splenic vein. Notice it's blue, carrying venous blood from the spleen towards the liver. So there's the splenic vein. Okay. Then there's the inferior mesenteric vein. Remember, the mesentery helps hold the intestines in place. So here is the inferior mesenteric vein. It merges with the splenic vein. Then the superior mesenteric vein, where's my cursor? Here we go, is on this side. Superior mesenteric vein. And then where these all merge together, you have a fairly large hepatic portal vein there, heading right into the liver. And then those nutrients are processed in the liver, checked for toxins, that sort of thing. And then draining the liver are the hepatic veins, as you see here. And they empty into the inferior vena cava, and then that carries venous blood to the heart. So there's a little diagram here that I often call the hepatic portal system chair. So if you think of a chair, you would sit on the flat part. Those are the front legs and back legs. And then here's the backrest of the chair where your back would recline against, okay? So what you're sitting on is the hepatic vein. Notice kind of cuts across, straight. And then coming down anteriorly, in terms of the chair, but in this case to the individual's left side would be the inferior mesenteric vein, the front legs of the chair, right there. And then the back legs of the chair, superior mesenteric vein, would be back here. In the sense of the back legs of the chair, but on your right side. And then the back of the chair is the hepatic portal vein. So it's kind of not exactly chair-like, but if you think of the hepatic portal system chair, that might help you get the veins in order, or help you identify the veins. Okay, some of the internal structure of the liver. So the liver is divided into what are called lobules. And these are the structural and functional units of the liver. The hepatocytes, the liver cells, right, hepatoliver-cyte cells, are arranged in these rows that are radiating out from the center, where you have a central vein right there. So the hepatocytes kind of radiate outwards, like spokes on a wheel almost. So blood from the hepatic portal veins and the hepatic artery branches here, they work through, I was saying, the hepatic portal veins and the hepatic artery branches in red, hepatic portal veins here in blue. They have blood vessel extensions that branch through the lobules into what are referred to as sinusoids, as you see here. So sinusoids are nice leaky blood vessels, these thin capillaries, so that the nutrients can be exposed and readily available to the hepatocytes for processing. Okay? And then the blood will percolate towards the central vein. Okay? Both arterial blood, essentially coming off of the abdominal aorta, and venous blood coming from the intestines, the spleen, and the blood would meander, percolate towards the central vein. So the hepatocytes, as I mentioned, they absorb, process the nutrients, they do produce bile, and the other functions that they'll do. These are the important ones to know. Now the bile that gets produced will get secreted from the hepatocytes and enter into the small bile vessels, referred to as bile canaliculi, little canals for bile. And the bile percolates in the opposite direction and leads to these interlobular bile ducts. There's also phagocytic immune type cells called Kupffer cells that are phagocytic. And that's these little pink ones that you see here, which are referred to as stellate macrophages here, but our textbook refers to them as Kupffer cells. Here's another picture I found of a section of a lobule that maybe gives you a better view of or sense of the venous blood flow direction, arterial blood flow direction, hepatocytes processing all those materials and nutrients, etc. Venous blood going towards the central vein, eventually going towards the hepatic veins, being returned to the inferior vena cava. And then notice in the opposite direction is the movement of the bile, also produced by the hepatocytes, heading towards these small blood vessels called bile ductules here, over here called bile duct. Okay? So at these regions along the boundaries of the lobules, they're referred to as portal triads because they have three components. Hepatic portal vein component here in blue, hepatic artery component in red, and then the bile duct in green. So 1, 2, 3, 1, 2, 3, 1, 2, 3. Those are the portal triads, right, of the liver lobules within the liver. And then bile secretion and transport. Hepatocytes produce the bile. Bile percolates through the bile canaliculi, through the bile ducts within the portal triad, and then eventually to the gallbladder for storage, and then eventually secretion into the duodenum, small intestine, as we talked about earlier. Okay? So, bile secreted from the hepatocytes and the lobules. Eventually, the bile will all come into one of the hepatic ducts, left or right hepatic duct. Then the bile gets stored into the gallbladder through what's called the cystic duct here for storage. And then when you are eating a meal and the food nutrients is leaving the stomach, entering into the duodenum, the bile gallbladder contracts, bile is ejected out through the cystic duct, out through the common bile duct, or just the bile duct here. And then the bile duct merges with the pancreatic duct, or the main pancreatic duct, at the hepatopancreatic ampulla, where the bile is then secreted into the duodenum out of the duodenal papilla. Okay? So the gallbladder is storing and concentrating bile, which is again used for lipid emulsification in the duodenum of the small intestine. As I mentioned, left and right hepatic ducts forming the common hepatic duct. The cystic duct leads to the gallbladder, and then the bile duct is leading towards the hepatopancreatic ampulla and then towards the duodenum. Okay? And the opening of the hepatopancreatic ampulla, the opening into the duodenum, is referred to as the duodenal papilla. The pancreas. Remember the pancreas, a mixed gland, posterior to the stomach, slightly inferior to it. Structurally, the head of the pancreas is at the duodenum, then there's the body, and the tail is out near the spleen. Again, a mixed gland has endocrine and exocrine function. Remember the endocrine function, the endocrine part, the pancreatic islets, or islets of Langerhans, are secreting these hormones, insulin and glucagon, that are regulating blood glucose levels.